Welcome to springlib.
Point mutations. So mutations are variations in the nucleotide sequence of a genome, and they can also occur because of damage to the DNA. So first we're going to look at these point mutations, and there are 3 different types. And point mutations are specific in that those mutations affect a single base pair. So here we're going to look at how a normal protein would arise. So we have the 5' and 3' end up here. This is our template. So when you look at this one, it matches the 5' to 3' end and the 3' to 5' end. So you have the TAC, ATG, that codes for the mRNA UAC, which is tyrosine, and that's going to encode a normal protein. So in this example, there's no mutation. All right, and then next we're going to look at this one here, the second one, where it normally would be TAC, it is now TAT, and as a result, you're going to have ATA going 3' to 5'. So your mRNA is going to be UAU, which is tyrosine again, and so in this example, this would be a silent mutation. And this is because there's no effect on the protein sequence, because you're going to encode a normal protein. All right, the third example here, instead of TAC, you have AAC, and then you're going to have TTG in the 3', 5' direction. That's going to encode for an mRNA that is AAC, which is asparagine, and that's going to be a faulty protein. So as a result of that, you're going to have an amino acid substitution, so tyrosine should be what it encodes for, but it's going to be asparagine. So in this example, it is a missense mutation. And then lastly, instead of TAC, you have TAG in the 5', 3', and then you have ATC instead of ATG in the 3', 5'. That's going to encode for the mRNA of UAG, which is a stop codon. And so that's going to result in an incomplete protein, and that's because the stop codon was substituted for the ideal amino acid, which is tyrosine. So this is going to be a nonsense mutation.